IGEM IIT Madras welcomes you to this video on translation. In the previous videos, we learnt about the revolutionary double helix structure for DNA. Very soon, Francis Crick proposed the central dogma in molecular biology, which states that genetic information flows from DNA to RNA to protein. During replication and transcription, a nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of complementarity. The process of translation, however, requires transfer of genetic information from a polymer of nucleotides to form a polymer of amino acids. Neither does any complementarity exist between nucleotides and amino acids, nor could any be drawn theoretically. There exist ample evidences, though, to support the notion that change in nucleic acids, that is genetic material, was responsible for change in amino acids in proteins. This led to the proposition of a genetic code that could direct the sequence of amino acids during synthesis of proteins. George Gamow, a physicist, argued that since there are only 4 bases, and if they have to code for 20 amino acids, the code should constitute a combination of bases. He suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. This was a very bold proposition because a permutation of 4 into 4 into 4 would generate 64 combinations, generating many more codons than required. Hargobind Khorana, Marshall Nirenberg, and Robert Hawley solved this proposition. They were awarded the Nobel Prize in Physiology or Medicine for the interpretation of the genetic code and its function in protein synthesis. The chemical method developed by Hargobind Khorana was instrumental in synthesizing RNA molecules with defined combinations of bases, that is, homopolymers and copolymers. Marshall Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. This proved that the codon was a triplet. Now, we know that the codon is a triplet and 61 codons code for amino acids and 3 codons do not code for any amino acids and hence function as stop codons. AUG functions as a start codon and also codes for methionine. The code is nearly universal. For example, from bacteria to humans, the codon UUU would code for the same amino acid, that is phenylalanine. The above two are just 2 of the many features of the genetic code defined and then proven to exist. We will discuss more interesting properties in the next video. Now, let's learn about translation. Translation refers to the process of polymerization of amino acids to form a polypeptide. The order and sequence of amino acids are defined by the sequence of bases in the mRNA. The amino acids are joined by a peptide bond. The cellular factory responsible for synthesizing proteins is the ribosome. The ribosome consists of structural RNAs and about 80 different proteins. In its inactive state, it exists as 2 subunits, a large subunit and a small subunit. When the small subunit encounters an mRNA, the process of translation of the mRNA to protein begins. There are two sites in the large subunit for subsequent amino acids to bind to and thus be close enough to each other for the formation of a peptide bond. A translational unit in mRNA is a sequence of RNA that is flanked by the start codon AUG and the stop codon and codes for a polypeptide. An mRNA also has some additional sequences that are not translated and are referred to as untranslated regions or UTRs. The UTRs are present at both 5' end before the start codon and at the 3' end after the start codon. These are required for efficient translation process. For initiation, the ribosome binds to the mRNA at the start codon AUG that is recognized by only the initiator tRNA. The ribosome proceeds to an elongation phase of protein synthesis. During this stage, complexes composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA anticodon. The ribosome moves from codon to codon along the mRNA. Amino acids are added one by one and translated into polypeptide sequences directed by DNA and represented by the mRNA. At the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. Thus, the transfer of information from DNA to RNA to protein is complete. In the next video, we will see how changes in the genetic code can cause mutations. Until then, goodbye.